and my project is on the effect of MSH2 gene on glioblastoma. Introduction. Glioma is a type of brain tumor that forms in glial cells. These cells are found around neurons and outnumber neurons 3 to 1. The purpose is to maintain homeostasis, form myelin, and provide support and protection for neurons. Glioma is classified within 4 different grades based on the tumor's potential to spread. Grades 1 and 2 are referred to as low grade, while 3 and 4 are referred to as high grade. Oncogenes are a group of genes that have the potential to cause cancer. They are formed during cellular replication when mutations occur. Genetic mutations are of many types, like single gene mutations, insertion deletion mutations, point mutations, and mismatch mutations. The primary role of tumor suppressor genes is to keep the cellular replication cycle in check. They tell cells to slow down, fix errors, and tell them to perform apoptosis. Mismatch mutations occur when the wrong DNA molecule is copied. For example, adenine instead of guanine. The gene I am focusing on for this project is MSH2. MSH2 is a tumor suppressor gene involved in DNA repair primarily. The diagram shows the MMR pathway, which is a DNA repair mismatch pathway. It is mostly used to repair base-based mismatches and insertion deletion errors. The aim of my project is to study the effect of MSH2 expression on glioblastoma using publicly available data sets. Glioblastoma is extremely important to study because although it is one of the most deadliest tumors, not very much is known about it at all. A hypothesis is that there will be low MSH2 expression in glioblastoma patients. My reasoning behind this is because of MSH2's key role in the mismatch repair pathway. If there are low MSH2 expressions, the MMR pathway won't function properly. I hypothesize that this will then lead to glioblastoma in the patients. I started off this project by learning about it, and I used it to design my hypothesis along with background research. The second step was to use bioinformatic tools. I used the National Cancer Institute, National Center for Biotechnology Information, PubMed database, and etc. to perform background research on both glioblastoma and MSH2. Next, I used the SNP and gene databases. These tools helped me learn in specific about the HMSH2 gene. Next, I obtained the data. I got the experimental data from the Cancer Genome Atlas, and there were a total of 607 individuals. Then I used cBioportal to obtain the clinical data for the patients. I had to match the experimental data to the clinical data. I also sorted the data for information that was relevant for my project. Relevant data was extracted into a separate file. Next, the RStudio program was used to perform an in-depth statistical analysis of MSH2 on FGA. I used correlation tests, t-tests, and survival analysis tests results. This data was obtained from the TCGA Firehouse Legacy dataset. The original sample size I started with was 607. I then had to match my clinical data to the experimental data. Not all my patients had data, so the number reduced to around 400. Next, I removed patients who had incomplete data, such as not having an FGA recorded. This table shows the statistics of the data. The mean age of the data set was 58. There are 178 people below 58 and 204 people above. There's a significantly higher amount of males in the study at 235 versus only 147 for female. Their mean ages are similar, however. Next, I performed t-test. A t-test is used to compare the means of two groups of data against each other. On this slide, I compared FGA versus age and sex. The first graph shows FGA versus sex. The mean FGA of female is about 0.24. The mean FGA of male is about 0.21. The p-value is less than 0.05, which means that the FGA in women is statistically significant to be different than in men. This slide has the MSH2 expression box plot. I split the MSH2 value into two groups based on a median of 0.06255. On this slide, there's a scatter plot that shows the correlation between FGA and MSH2. The R value represents how strong the correlation is between both variables. The R value here is 0.14. This shows that there's a positive correlation, although it is not very strong. This is due to many outliers right here, that, as can be seen on the graph. On this slide, there are the Kaplan-Meier survival analysis plots. Kaplan-Meier plots are used to measure the amount of time a person is living after treatment. The first plot shows the MSH2 on disease-free status, and the second plot shows MSH2 versus OSS. These tests show that there is no significant effect of MSH2 on DFS or OSS, and this completes my third objective of analyzing survival data. My hypothesis was proven to be rejected because the data was sorted based on my objective. Throughout my analysis, I discovered that there is a significant positive correlation between MSH2 and FGA. In the future, I would like to perform a more in-depth analysis and research about the role of other genes on glioblastoma.
What do you mean by 1 and 2? And then how is the age there? What kind of age groups were there, and how you doing this? It was not clear. Could you explain that again?
Okay, so I split the age groups into based on the mean. The mean was 58. So age group 1 would include means of 58 and below. Age group 2
Okay.
would be 50, above 58.
Okay. All right. And then so it looks like um MSH2 is more in the old population. Is that correct?
That's correct.
Okay. All right. Okay. Yeah. And then can you go to the next slide just quickly? I think I had one more question on the next slide, but yeah, this one is again the same groups you were talking about, the 58 or
This one is split based on the median value of the MSH2. So below
okay.
the MSH2 of the median of 0.06255 is
Oh,
1, above is 2.
okay. All right. Okay. Because it wasn't clear to me. Thank you so much. Thank you.
Thank you.
Great presentation. As mentioned, you can improve it by adding the slide numbers. This helps for people to make notes. And then I would also add to improve your next presentation, you can when you present the numbers, you can stop at some significant digits when you give 0, 0, 5, not 0, 0, 5, 6, 7, 8, 9, 10, and so on, you know. Uh, and I would say it's great to see people doing and confidently presenting negative data because what you found, your hypothesis is not working, it's perfect, but in real life, that would be very hard to present only the negative data. So it's good to think further to find something that uh, uh, would be a difference that you found. After all, great job anyway. Thank you.
Thank you for the feedback. Thank Newton and Dr. Manuel for this opportunity. And I really enjoyed this experience, and I'm going to continue to enjoy it when I'm continuing Dr. Manuel.